Our new lecture is about the biochemical basis of chronic obstructive pulmonary disease and smoking. In this lecture, we have some questions about elastin. What is elastin? What is alpha-1 antitrypsin? What is the effect of smoking and what is the mechanism by which it acts? So we will start with elastin structure, function, and the protective role of alpha-1 antitrypsin in counteracting the protease activity. So to answer our first question, elastin is one of the fibrous and elastic proteins that is present abundantly in tissues that need elasticity as well as tensile strength, as it can endure stretch. Chemically, it is composed of 4 polypeptide chains in the form of a tetramer, similar to collagen in structure, but it has 33% of the amino acid components of glycine and is rich in proline, but it has low hydroxy proline with absence of hydroxylysine. So, as we mentioned, that elastin fibers can be stretched to several times as its normal length and is present in the walls of large blood vessels such as aorta. It is very important in lungs, elastic ligaments, skin, and cartilage. Defects in the structure of elastin are associated with defective function, like what occurs in emphysema, which is an example of chronic obstructive lung diseases and result from defective elastin. So for elastin as one of the extracellular matrix proteins, it may be subjected to proteolytic destruction, and this demands protection. So the protective agent is the alpha-1 antitrypsin or alpha-1 antiproteinase, which is a protein involved in protecting elastin, collagen, and other extracellular proteins from the destructive effect of proteases activated during various disease states. So what is alpha antitrypsin or alpha-1 antiproteinase? Alpha-1 antiproteinase is a protein of a molecular weight about 52 kilodaltons, and it was formerly called alpha-1 antitrypsin. It is a single chain protein of 394 amino acids that contain 3 oligosaccharide chains. This protein constitutes the major component, more than 90% of the alpha-1 fraction of human plasma. This protein is synthesized by hepatocytes and macrophages and is the principal serine protease inhibitor of human plasma. Regarding the clinical importance, there are 2 clinically interesting areas concerning alpha-1 antitrypsin deficiency, one of which occurs in approximately 5% of cases of emphysema, and this occurs mainly in the homozygous and heterozygous subjects who synthesize and secrete considerably less protein than normal individuals. The other condition is implicated in 1 type of liver diseases, which is alpha-1 antitrypsin deficiency liver disease. The question is, what is the role of alpha-1 antitrypsin? Alpha-1 antitrypsin is an enzyme secreted from the liver, as we mentioned before. It's responsible for inhibiting elastase, and thus it inhibits elastin destruction and provides a protective role for elastin and other extracellular matrix proteins. And as we know that elastin is one of the extracellular matrix proteins present in lungs, blood vessels, and there is deficiency of alpha-1 antitrypsin, especially in chronic smokers. Smoke inhibits the alpha-1 antitrypsin, and thus elastin of the extracellular matrix is not protected, and especially in the lungs. And elastase thus is working or acting unopposed and causes destruction of elastin of the lungs, leading to loss of elasticity of lung tissue with resulting emphysema, which is chronic obstructive lung disease, particularly in cigarette smokers. The mechanism of action of alpha-1 antitrypsin. This enzyme inhibits proteases like trypsin, elastase, and certain other proteases by forming complexes with them. In its structure, alpha-1 antitrypsin has a particular methionine residue at the position 358 of the amino acid sequence of the protein, which is involved in binding to proteases. So the methionine 358 is an essential residue for binding elastase. Deficiency of the alpha-1 antitrypsin is also implicated in a type of liver disease, as mentioned, the alpha-1 antitrypsin deficiency liver disease, and also by mechanisms that are not understood. Hepatitis results with consequent cirrhosis, and in this case there is accumulation of massive amounts of collagen resulting in fibrosis, and this is one of the conformational diseases of the liver. Other members of this group of disorders, the conformational disease, include Alzheimer's disease, Parkinson's disease, and Huntington disease. So the question now is, what happens in cases of chronic smoking and inflammation? When the amount of alpha-1 antitrypsin is deficient, or when polymorphonuclear white blood cells increase in the lungs, for example during infection, especially during pneumonia and other infections of the respiratory system, affected individuals lack the counter-shock mechanism to the proteolytic damage of the lung by elastase. Smoking also as one of the risk conditions. Smoking oxidizes the 358 methionine residue to methionine sulfoxide, and by this it inactivates the alpha-1 antitrypsin. As a result, affected molecules of the alpha-1 antitrypsin lose their protease neutralizing action. This problem is particularly devastating in patients with the homozygous phenotype who already have low levels of alpha-1 antitrypsin. And further reduction in alpha-1 antitrypsin brought about by smoking results in increased proteolytic destruction of lung tissue, and this will accelerate the development of emphysema. But what about infection and inflammation? And what occurs in these cases? When there is infection or inflammation, there is recruitment of phagocytes and macrophages to the site of infection or inflammation, and phagocytosis of microorganisms, especially bacteria and foreign particles, occurs as a defense mechanism during infection. And as you know, macrophages have an oxygen-dependent system, which is a myeloperoxidase system, which uses the reduced NAD phosphate oxidase and the white blood cell membranes that converts oxygen into superoxide free radicals. This superoxide is converted by superoxide dismutase into hydrogen peroxide. Lysosomal myeloperoxidase converts this hydrogen peroxide into hypochlorous acid, which kills bacteria. So conditions of deficiency of the NADPH oxidase are also associated with chronic granulomatosis and chronic lung disease. The increased amounts of these chlorinated oxidants as hypochlorous acid that result during infection and inflammation also affect the proteinase antiproteinase equilibrium. So this tilts the balance in favor of the proteinase with consequent tissue damage. These oxidants are not the only factors that activate proteinases, but there are other factors and other proteinases that are listed as you see in the following table. This table shows some examples of proteinases and antiproteinases present in plasma and tissues. And we have elastase and collagenase and their antiproteases or antiproteinases. We have the alpha-1 antitrypsin as an example. So the chlorinated oxidants, as we mentioned, that result during inflammation, and the antioxidant activity exerted by the glutathione defense system with invading microorganisms, and in production of the chlorinated oxidants. And these destroy microorganisms but activate proteinases present in extracellular matrix and inactivate the antiproteinases. So they have beneficial and destructive effects as well. To know how proteolytic damage of the lung occurs by elastase, we can check the figure to see what occurs in normal and in disease conditions. And as you see in the figure, during normal conditions, neutrophils release elastase, but with the presence of alpha-1 antitrypsin, elastin is protected with no damage to tissues. But during inflammation, elastase acts unopposed due to inactivation of the alpha-1 antitrypsin by chlorinated oxidants, for example, ending in destruction of lung tissue or any other elastin or extracellular protein present. So the sequence of events will be when there is active elastase and active alpha-1 antitrypsin. This inactivates elastase with no proteolysis of tissue. But when the active elastase acts unopposed due to inactive or inactivation of alpha-1 antitrypsin, the activity of elastase results in proteolysis of tissue and end in tissue damage. For the treatment of these cases, there are different lines of treatment. Some cases are treated with weekly doses of alpha-1 antitrypsin, intravenous administration of alpha-1 antitrypsin in patients with emphysema due to the alpha-1 antitrypsin deficiency. Also attempts to generate alpha-1 antitrypsin mutants by genetic and protein engineering. These mutants are not inactivated by any factor, and there is replacement of the methionine 358 by another residue that is not subject to oxidation. Also, attempts for gene therapy are present, but this is of course very, very costly. At present, some severe cases of alpha-1 antitrypsin deficiency liver disease can be successfully treated by liver transplantation. These are the main lines of treatment of chronic obstructive pulmonary disease due to alpha-1 antitrypsin deficiency. If you like this lecture, please hit the subscribe button and give your feedback in comments. The link for this lecture will be in the description below this video, and you can find other details in the website provided.